Hi everyone, welcome back to the channel The Science of Time. This is Pratikshya Paul, a third year MBBS student, and this is yet another video in my histology series, and today we are going to discuss about the male reproductive system. So without any further ado, let's get into the video. So, in the male reproductive system, we are going to discuss 4 slides today. And these slides are of testis, epididymis, vas deferens, and prostate. And these are the important slides of male reproductive system and often comes in your first MBBS final exam. And so, let's start discussing about the first slide, that is the slide of testis. So, what is the capsule of testis? It is consist of 3 layers. From outward to inward, it is tunica vaginalis, tunica albuginea, and tunica vasculosa. So you have to remember it from outward to inward or inward to outward manner, whichever do you like. So uh, the capsule of testis consists of 3 layers. And what do you see in the cut section of testis? A number of seminiferous tubules cut in various directions. The tubules are separated by connective tissue containing blood vessels and Leydig cells, and each tubule is lined by 2 types of cells. One is spermatogenic cell that produce the spermatozoa, and second is the Sertoli cell that provide the supportive function or the nutrition to the developing spermatozoa. So, what are the important characteristic features of the slide of testis? You will have to identify a number of seminiferous tubules that are cut in various directions, and the tubules are separated by the connective tissue. You may or may not find the Leydig cells, and the capsule of testis you may find or you may not find because it is not present in every slide. Also, you have to identify the 2 types of cells that are present in each seminiferous tubule, that is the spermatogenic cell and the Sertoli cell. Okay, now come to the slide. So here you can see these are the cut section of the seminiferous tubules present. These are the cut section of seminiferous tubule, and these are the connective tissue present between the seminiferous tubules. These are the connective tissue present between the seminiferous tubules, and this is the real slide picture. You can easily identify the uh, seminiferous tubules present in the cut section, and you can see these are the spermatogenic cell present towards the periphery. These are the spermatogenic cell, and towards the lumen, these are the Sertoli cells present in the seminiferous tubule. And also towards the periphery, the first cells that are towards the periphery are the primary spermatozoa. Then below that, these are the secondary spermatozoa, and after that, the immature and mature sperms are present. So towards the lumen, they are the mature sperms present. Okay, so now you can easily identify the slide of testis. So this is the picture from your diffuse atlas. Here the capsule of the testis is present, but it may not present in every slide. So you have to identify the cut section of the seminiferous tubule and the cells that are the spermatogenic cell and the Sertoli cell in the tubule. And these are the important characteristic feature of your slide of testis. So you have to draw this diagram in your copy, and you have to write the identification features. Okay, this much about the slide of testis, and now let's go to the second slide, that is the slide of epididymis. So what are the characteristic features of epididymis? The tubules are lined by pseudostratified columnar epithelium bearing stereocilia, and the epithelium is composed of 2 types of cell, basal cell and principal cell. So what is the difference between basal cell and principal cell? Basal cell is short and do not reach the lumen. It have rounded nuclei and the nuclei stands dark. Whereas the principal cells are tall cells, it has irregular oval nucleus and the nuclei stands light. Okay, so this is the characteristic feature of epididymis. There are no more features. So you have to remember the pseudostratified columnar epithelium and the presence of 2 types of cell in the epithelium, that is the basal cell and the principal cell. And from these 2 cells only you will identify the slide of epididymis. Okay, so now let's come to the slide. So can you see these are the tubules of epididymis present? These are the tubules of epididymis, and you can easily identify the basal cell and the principal cell from its staining of nucleus, from its nucleus only. So you can see these are the darkly stained nucleus. The darkly stained nucleus are of the basal cells. So these are the basal cells. These are the basal cells. And towards the lumen, you can see these are the lightly stained cells. These are the principal cells present. These are the principal cells. And in the lumen, these are the mature sperms present. These are the mature sperms, because it is the function of the epididymis to store the mature sperms. So in the lumen of the tubules of epididymis, these are the mature sperms, and towards the lumen, can you see these small protrusions? These small protrusions are, these are the stereocilia present. So what is the function of the stereocilia? The stereocilia propel the sperms throughout the epididymis, throughout the tubules of the epididymis. Okay, now you can easily identify the slide of epididymis. This is the real slide picture of epididymis, but it is a low power view, so you can only identify the cut section of the tubules of epididymis, but you cannot identify the basal cells and principal cells here. I do not have a better picture for epididymis. So you can easily identify the slide of epididymis from these features only. This is the picture from your diffuse atlas. You have to draw it in your practical manual. Here also you can see in the lumen mature sperms are present. This is the pseudostratified epithelium, and the dark stained nuclei are the basal cells, and the lightly stained nucleus are the principal cells. Okay, and the stereocilia is also towards the lumen, help in propelling the sperm. Okay, so this much about the slide of epididymis. अभी तुम्हें कभी कभी लग सकता है कि स्लाइड ऑफ एपिडिडाइमिस एंड टेस्टिस तो काफी सिमिलर है, एक जैसा ही लग रहा है, दोनों में कट सेक्शन ऑफ टिब्यूल्स ही तो है. But it is very easy to differentiate it between the slide of epididymis and the testis, because in testis there was no epithelium. In epididymis, pseudostratified epithelium is present, which you can easily differentiate, and also the basal cell and principal cell you can easily differentiate by the property of their nucleus. Okay, so this much about the slide of epididymis. Now let's come to the slide of vas deferens, or it is also known as ductus deferens. It is a tubular structure. The other tubular structure that you can find as a slide are of ureter, bladder और appendix. So अगर तुम्हें स्लाइड में कोई ल्यूमेन दिख रहा है तो तुम्हें इजीली पता चल जाएगा कि इसमें से ही कुछ होगा. तो तुम्हारे पास 3-4 स्लाइड की तुम 3-4 स्लाइड के बारे में सोचना चाहिए, बिकॉज बाकी सब तो रूल आउट हो गए. Okay, so what are the characteristic features of vas deferens? It is a tubular structure. It has a small irregular lumen. The mucous membrane is lined by pseudostratified columnar epithelium. The lamina propria is present beneath the mucous membrane. The muscle coat is very thick. It contains 3 layers: the inner longitudinal layer, the middle circular layer, and the outer longitudinal layer. Okay, so there are 2 longitudinal layers, the inner and outer, but the middle is a circular muscle layer. And the adventitia is composed of collagen fibers and blood vessels. Okay, so what are the characteristic features of vas deferens? You have to identify the pseudostratified columnar epithelium in the lumen, and you will find a lumen. That is the most important thing. You have to identify lumen होगा slide में, और in the mucous membrane it will have pseudostratified columnar epithelium, and another important feature is the muscle coat, because the muscle coat of vas deferens is very thick. इतना थिक मसल कोट तुम्हें और किसी स्लाइड में नहीं मिलेगा. ओके, सो यू विल इजीली आइडेंटिफाई फ्रॉम these 3 points, that is the muscle coat, the lumen, and the epithelium. Okay, so now let's come to the slide. So this is the pseudostratified columnar epithelium present. This is the lamina propria beneath the mucosa, and this is the muscle coat present. Okay, this is just a cross section, and this is the lumen that you can see, and the mucosa contains pseudostratified columnar epithelium, and beneath the mucosa it is the lamina propria, and behind it is the muscle coat. Okay, you can easily identify it from the lumen and the muscle coat and the epithelium present. This is the real slide picture. You can identify this is the irregular lumen present, and this is the muscle coat present. It is a low power view, so you cannot easily identify the circular and longitudinal muscle layer, but in under the microscope you can identify the circular and longitudinal muscle layer and differentiate it, but it is not that important. You have to identify the slide and write the characteristic features only. So from the irregular lumen and the thick muscle coat present, you will easily identify and differentiate it from the ureter or bladder. We will discuss in the next session about the urinary system. There I will again tell you about the differentiating features of vas deferens and ureter or bladder. Okay, and this is the picture from the diffuse atlas. You have to draw it in your practical manuals. Okay, so this much about the slide of vas deferens. You can easily identify the slide of vas deferens now. So let's come to the last slide of this session, that is the slide of prostate. So what are the characteristic features of prostate? Prostate is the largest accessory sex gland. It is a fibromuscular glandular organ. Means fibromuscular stroma is present and the glandular tissue is also present. So the prostate consists of the glandular tissue embedded in the prominent fibromuscular stroma. The glandular tissue is in the form of follicles with serrated edges. You can easily identify the serrated edges. The glandular tissue lined by columnar epithelium, and the lumen may contain amyloid bodies or corpora amyloacea. And the follicles are separated by the fibromuscular tissue. So these are the important characteristic features of prostate. You have to identify the glandular tissue and in the fibromuscular stroma, and the glandular tissue is in the form of follicles with serrated edges which you can easily identify under the microscope. Okay, and the lumen contains the amyloid bodies or corpora amyloacea. There may not be corpora amyloacea in every lumen, but you can identify it easily. Okay, now let's come to the slide. Okay, so these are the glandular structures present. You can easily identify these are the follicles present and these are the serrated edges. Can you see? Can you appreciate the serrated edges present? These are the serrated edges present in the towards the lumen. Okay, and this is lined by the columnar epithelium. The glandular part is lined by the columnar epithelium. You can easily identify it under the microscope. Okay, here the fibromuscular part is not present in this slide, but you can identify it from here. These are the glands present. This is the glandular tissue. This is the glandular tissue present, and between the glandular tissue, this is the fibromuscular organ present. Sorry, fibromuscular coat. Okay, so you can here also identify the serrated edges of the follicles, and the glandular tissue is composed of columnar epithelium. This is the real slide picture. You can easily identify this is the glandular part, this is one glandular part, and between the two glandular part, it is the fibromuscular coat present. Okay, and this is the picture from your diffuse atlas, but in this picture there are prostatic urethra and prostatic sinuses ejaculatory ducts, but you may not identify these in the slide, or it may not be present in every slide also. So you have to just identify the glandular tissue and the fibromuscular coat. That is enough for you to identify the slide as a prostate. And also I forgot to tell you this, you can see these are the uh, amyloid bodies that are present in the lumen. These are the amyloid bodies present in the lumen of the follicles. So it is not present in every lumen, but it is present in some lumen. So you can easily identify it from that, because अगर तुम स्लाइड को प्रोस्टेट आइडेंटिफाई कर रहे हो तो अगर उसका ल्यूमेन में कुछ है तो वो amyloid बॉडी ही होगी. और ओके, so now you can easily identify the slide of prostate from the glandular structure, the fibromuscular coat, and the follicles are lined by columnar epithelium with serrated edges. This is the 4 points that you need to identify the slide as a prostate. Okay, so this much for today's session. I have discussed the 4 slides of male reproductive system: testis, epididymis, vas deferens, and a prostate. In my first year final exam, I have the slide of testis and I have identified it, but वहाँ पे मुझे confusion हुआ था कि ये testis है या epididymis है, because फर्स्ट ईयर में हमें इतना पता नहीं चलता है, बट हिस्टोपैथोलॉजी पढ़ने के बाद मुझे हिस्टोलॉजी का अंडरस्टैंडिंग इतना अच्छे से समझ पाई हूँ मैं हिस्टोलॉजी को, बिकॉज अगर तुम नॉर्मल और एबनॉर्मल दोनों पढ़ लोगे ना तो तुम्हें इजीली आइडेंटिफाई कर पाओगे कि एक किस चीज़ का स्लाइड है. बट स्टिल आई हैव डिफिकल्टी इन आइडेंटिफाइंग प्रोस्टेट एंड सिक्रेटरी एंडोमेट्रियम इन सेकेंड ईयर ऑल्सो, बिकॉज दोनों काफ़ी सिमिलर लगते हैं. बट इवेंचुअली वी विल गेट थ्रू इट, बिकॉज तुम वही बार बार वही चीज़ देखोगे तो वो चीज़ तुम्हें आ जाएगी एट द एंड. Okay, so I think I have discussed everything important in respect to the male reproductive system histology. That's it from my side for today. Thank you so much for watching, and you can ask your doubts in the comment section. I will see you in the next session of histology. Till then, keep studying, keep watching.